Hello everyone, welcome back to my channel. This video is about monoclonal antibody production. Definition: monoclonal antibodies are the antibody that are made by identical immune cells that are all clone of a unique parent cell. Monoclonal antibody can have monovalent affinity in which they bind to the same epitope. This technique was devised by Georges Kohler and Cesar Milstein in 1975. Method or the steps that are used for the production of monoclonal antibodies are immunization of a mice, isolation of B cell from the spleen, cultivation of myeloma cells, fusion of myeloma and B cells, separation of cell lines, screening of suitable cell lines, in vitro or in vivo multiplication, lastly harvesting. From this picture we can see a mice is injected with an antigen and we are isolating spleen cells from the mice, whereas myeloma cells are cultured. Myeloma cells known as cancer of a plasma cells. Now spleen cells and myeloma cells are fused together and we are growing only hybrid cells in a hat medium. Hat medium contain hypoxanthine aminoprotein thymidine. It is a selection medium used for mammalian cell culture. Now we are separating the hybrid cells and allow them to proliferate into clone, which means we are multiplying it, known as hybridomas. Screen for desired antibody. We are screening the required antibody for the production. Now harvest the monoclonal antibodies in larger amount. Production of monoclonal antibody. Monoclonal antibodies are produced in vitro condition using tissue culture technique. In vitro condition is nothing but under lab condition. A mouse is injected with specific antigen that will induce antibodies against that antigen. Now the spleen cells of the mouse is removed and homogenized into a cell suspension. The suspension includes B cells that produce antibody against the injected antigen. The spleen cells are then mixed with myeloma cells, cancerous B cells that are capable of continuous growth in culture but have lost the ability to produce antibodies. Some of the antibody producing spleen cells and myeloma cells fuses to form hybrid cells. These hybrid cells are now capable of growing continuously in culture while producing antibodies. The mixture of cell is placed in a selective medium that allow only hybrid cells to grow. Hybrid cells proliferate into clones called hybridomas. The hybridomas are screened for the production of desired antibody. The selected hybridomas are then cultured to produce large quantities of monoclonal antibodies. This is a very expensive and time-consuming process. It may take weeks of culturing and many liters of media to provide enough monoclonal antibody for an experiment or to treat a single patient. Monoclonal antibodies are expensive. Applications of monoclonal antibody. These antibodies are used in various fields such as for diagnostic tests, Western blot, immunodot blot and immunofluorescence test. Analytical and chemical use for immunoprecipitation. It is also used for therapeutic treatment and also cancer therapy. These monoclonal antibodies are used for autoimmune disease. Thank you for watching this video. I hope it is very useful to you all. Don't forget to like, share, comment and subscribe my channel. Tada, bye bye.